And for all the parents at home, thank you very much for everything you did. Next is Yalda Shiwan, rising junior. Yalda's a rising junior at the University of Washington. Yalda, you ready?
Hello, everyone. Thank you for being here. So my name is Yalda Shiwan, and today I'm going to talk about my summer that I spent in Dr. Darvas' lab studying the role of GPA in Parkinson's disease. So to start off, I would like to outline some key point of my project, which is Parkinson's disease, also known as PD, role of GPA in Parkinson. But the main objective of this project is to investigate whether GPA deficiency indicates the propagation of alpha-synuclein pathology in brain. And we use the Preformed fibrils, which is also we know as PFF in mouse model. So as we all know, that Parkinson's disease is the second most neurodegenerative disorder after Alzheimer, and unfortunately, in fact, millions of people worldwide. And some of the motor symptoms for Parkinson's disease is the rigidity, tremor, and also slowness of movements. And these symptoms occurs to the propagation, loss of dopamine, which is protein in substantia nigra, a part of the brain. And it's also affects by accumulation of misfolded alpha-synuclein, which is a brain also known as Lewy body, and it's a hallmark of Parkinson's disease. And also the role of GPA. GPA is an enzyme that is called GCase, which is leading to the lysosomal dysfunction. And when the waste product built up, it's built up the misfolded alpha-synucleins, which is caused the Parkinson's disease propagation. So to figure out the propagation of Parkinson's disease, we use the Preformed fibrils or PFF, which is a synthetic alpha-synuclein fibrils generated in vitro or here on the striatum. And so we use the PFF in two type of the mouse, which is wild type and GBA heterozygous. And here we can see that the PFF has affect spread out all the brain. So the methodology that we used is we used the PFF and injected them on 4 months old mice, and then we let them to grow up until 10 months, and then we used the humanly euthanized, and we take out the brain. We embedded them at negative 80. After that, we slice them, and we use the antibodies to stain the tissue. And after that, we use the microscope to take the picture to see the spread of alpha-synuclein on the brain. So before I was doing the imaging, I wasn't told to know which one was the PFF injected and which one was not the injected one. So at the sun mark that I was looking, it was like this green stuff that it shows the a signal of PFF, and the other one is which is a control one. There is no signal. So here we can see on 510, which is on a striatum, a part of the brain that is, it shows the injected part. And here on 512 is hippocampus, that also we can see the green inclusion on the brain. And on 509, 515, we can see that it's spread out all the brain. So as a result, we found out that the PFF injected brains based on the histological marks that This green inclusion, it's a PFF alpha synuclein, which is built the Lewy bodies, also a hallmark in Parkinson's disease. So to go further, we can understand that GP deficiency really affects on alpha synuclein pathology in GBA heterozygous mice, and to study the real effect of GBA heterozygous, we can use this pathology. And I would like to thank Dr. Ellenbogen for giving me this opportunity to be part of this program, not just for me, for all other students, give the chance to learn and grow and put our skill in practice. And also Julie, my mentors, Dr. Davis, which is through the Zoom, and Dr. Martin Darvas, and also all the other people that was involved in this program. And last but not least, UW Medicine and IH Grant that make all this possible. Thank you so much.